Hello, my name is Isabella de la Paz, and today I will be presenting the research article titled TRBC2 Targeting Antibody Drug Conjugates for the Treatment of T-cell Cancers. Throughout this presentation, I will be referring to antibody drug conjugates as ADCs. Now, the main focus of this study was to develop a more selective treatment for T-cell cancers. The researchers wanted to treat malignant T cells without destroying all normal T cells because normal T cells are essential for immune protection. In this presentation, I will explain the goal of the study, how the researchers approach that goal, some of the major results, and the overall outcome and impact on future research. And these are all of the sections my presentation will cover in order to examine and explain aspects of this research article. T-cell cancers include T-cell leukemias and lymphomas, and they're difficult to treat because cancerous T-cells and healthy T-cells share many of the same surface markers. This creates a major challenge. If a treatment targets all T cells, it may kill the cancer cells, but it can also remove many normal T cells and cause dangerous immunosuppression. The article explains that normal T cells express either TRBC1 or TRBC2. In contrast, T cell cancers usually come from one clonal population, meaning that the cancer cells in a patient tend to express only one of these versions. So it expresses either TRBC1 or TRBC2. This creates an opportunity. If a patient's cancer cells express TRBC2, then a treatment could specifically target TRBC2 and still leave the TRBC1 expressing normal T cells behind, or vice versa, making TRBC1 or TRBC2 the selective targets. And this is basically the main biological idea behind the study. The goal of the study was to create a high affinity antibody that could specifically recognize TRBC2 and then use that antibody to build an antibody drug conjugate, or ADC. An ADC is basically an antibody attached to a toxic drug payload. The antibody acts like a delivery system because it recognizes the target on cancer cells, and then the toxic payload is brought into those cells to kill them. In this case, the authors wanted the ADC to kill TRBC2 positive T cell cancers while sparing TRBC1 positive normal T cells. So the goal was not just to kill cancer cells, but to do it selectively enough to preserve part of the normal immune system. So to approach this goal, the researchers and authors first engineered an anti-TRBC2 antibody using phage display. They started from a previously known anti-TRBC2 antibody and modified parts of the antibody sequence to improve its binding. They screened many antibody variants and looked for ones that bound strongly to TRBC2, but not TRBC1. After identifying the best antibody candidate, called JX1.1, they tested its binding using methods such as surface plasmon resonance and flow cytometry. Then they tested whether the antibody could actually be internalized into TRBC2 positive cells because internalization is necessary for an ADC to deliver its toxic payload. After that, they attached a cytotoxic payload to the antibody and tested whether the ADC could kill cancer cells in cell culture, patient-derived samples, and mouse models. So now on to some of the major results from this study. The first major result was the generation of the GX1.1 antibody. This antibody had strong affinity for TRBC2 and did not show meaningful binding to TRBC1, which is exactly what the researchers wanted. This matters because specificity is the key to making the treatment safer. If the antibody also bound to TRBC1, then it would lose its selective advantage and could damage a much larger portion of normal T cells. The authors compared JX1.1 with other anti-TRBC2 antibodies and found that JX1.1 had the strongest binding performance, which can be noted throughout panels A through Z in the Figure 1 image. This made the JX1.1 antibody the best candidate for further development into an ADC, since it was better than all the other antibodies that were previously tested. The second major result was that JX1.1 specifically bound TRBC2 positive cells. The researchers tested this using several T cell cancer cell lines. JX1.1 bound to cell lines that naturally express TRBC2, and it also bound to CRISPR-edited Jurkat cells, to the Jurkat cell line, which that cell line is also designed to express TRBC2. Importantly, it did not bind strongly to TRBC1 positive cells or cells lacking the T cell receptor. They also tested normal human T cells from healthy donors and found that JX1.1 could distinguish TRBC2 positive T cells from TRBC1 positive T cells. And this supported the idea that the antibody could selectively identify the target population instead of broadly binding to all T cells. So it is selective, which is another thing the researchers were looking for. Now, the third major result was that the antibody was internalized into the TRBC2 positive cells. This is important because ADCs need to enter the cancer cell to release their toxic payload. The authors used a pH-sensitive dye called pH-roto. This dye becomes fluorescent in acidic environments like lysosomes. When JX1.1 was added to TRBC2 positive cells, fluorescence increased over time, showing that the antibody entered the cells and reached lysosomes. In contrast, this increase was not seen in TRBC2 negative cells. The authors also confirmed lysosomal localization using LAMP1 staining. The staining and fluorescence can be seen in panels D and E in the Figure 3 image. And these results basically showed that JX1.1 was not only binding to the cell surface, but it could also be taken inside the cell, making it useful for ADC development. The fourth major result was that the JX1.1 ADC effectively killed TRBC2 positive cancer cells. The researchers attached a toxic payload called SG3249, which is a dimer payload that damages DNA and leads to cell death. They compared JX1.1 ADC with ADCs made from other anti-TRBC2 antibodies. And JX1.1 ADC showed much stronger cytotoxicity with a very low IC50 value, meaning only small amounts of the ADC were needed to kill TRBC2 positive cancer cells. At the same time, the ADC showed minimal toxicity toward TRBC2 negative cells. This result is important because it shows both potency and specificity. So far, all the results are what the authors were hoping for and looking for within their goals. Additionally, the authors also tested the JX1.1 ADC on patient-derived cancer cells from patients with cutaneous T-cell lymphoma or CTCL. This step is important because cell lines are useful, but patient-derived samples are closer to real disease. In these samples, the malignant T cells expressed TRBC2 and had loss of CD26, which helped the researchers distinguish cancer cells from normal T cells. The JX1.1 ADC selectively killed the TRBC2 positive malignant cells, while relatively sparing the TRBC1 positive normal T cells. This result strengthens the clinical relevance of the study because it showed that the treatment was not only effective in engineered or standard cell lines, but also in patient-derived cancer cells. And figure 5 from the article demonstrates some of the results from these patient-derived cells. The final major result I would like to discuss and highlight from this research article came from mouse models. The researchers tested JX1.1 ADC in mice carrying TRBC2 positive T cell cancer xenografts. They used bioluminescence imaging to track tumor burden over time. In the control ADC group, the cancer expanded, the mice lost weight, and disease progression led to worse survival. In contrast, mice treated with JX 1.1 ADC had reduced tumor burden, stable body weight, and improved survival. The researchers also tested another model using HPB ALL cells and saw similar results. They also tested patient-derived xenografts and found that JX1.1 ADC reduced circulating cancer cells, while control mice showed cancer expansion. All of these tests and results can be seen in the various panels in the figure 6 image. And overall, the mouse data supported the idea that this ADC could work in vivo, not just in cell culture. Now, the overall outcome of this study is that the authors successfully developed a high affinity anti-TRBC2 antibody and used it to create a selective ADC for TRBC2 positive T cell cancers. Why is this important? This is important because it fills a major gap in T cell cancer therapy. TRBC1 targeted approaches already exist, but patients whose cancers express TRBC2 also need a selective treatment option. The JX1.1 ADC could potentially become an off-the-shelf therapy, meaning it would not need to be custom made for every patient in the same way some cell therapies are. For future research, several steps are still needed. The antibody contains a marine FC region, so it would still need to be modified for clinical use in humans. Researchers will also need to study safety, dosing, possible infection risks, and possible relapse mechanisms. The article notes that relapse could happen if cancer cells lose TRBC2 or TCR expression, or if they develop resistance to the payload. Still, the impact of this study is significant because it shows that TRBC2 can be used as a selective therapeutic target. It also suggests that TRBC directed targeting could potentially be useful beyond cancer, including diseases driven by harmful T cell clones. Overall, this study provides a strong preclinical foundation for developing a new targeted therapy for patients with TRBC2 positive T cell cancers. These were all of the references I used for this presentation. And thank you so much for listening.